Hello, everyone. Thank you for tuning into my presentation. I'm Chad Teal. I'm a PhD candidate at the University of Arizona, studying fisheries conservation and management under Dr. Scott Bonar. My dissertation focuses on developing Trojan sex chromosome carrying red shiner and green sunfish to control their invasive populations that we have here in the Southwest. So 15 minutes is just not nearly enough time to go into any meaningful detail on this project. So if you have any questions about the research or this presentation in general, please feel free to reach me at chadteal@email.arizona.edu. So red shiner and green sunfish are two species that are considered highly invasive when introduced outside of their native range, and their invasive populations have been known to negatively impact populations of multiple species, many of which are of high conservation concern. So how can we effectively and selectively eradicate nuisance populations of these two species? One promising strategy is known as the Trojan sex chromosome strategy, where we release either YY males or ZZ females that can mate with wild type females and skew the sex ratio towards all male. So fish sex determination systems are very complex, and the variety is incredible. So in many species, the male is the heterogametic sex, like that in mammals, and in other species, the female is the heterogametic sex, like that in birds. But environmental factors and other mechanisms can deviate from these more traditional sex determination systems, although the majority of fish species do have chromosome-based sex determination systems. So in a species where the male is the heterogametic sex, you release males that are homozygous for the male coding region, and they will spawn with wild type females to produce all male offspring. In a species where the male is the homogametic sex, you produce homogametic females and or feminized males. They will spawn with wild type males and produce all male offspring, which will also skew the sex ratio towards all male. Through enough introductions of Trojan sex chromosome carriers and over a long enough period of time, you can skew the sex ratio towards all male and eradicate all the females from the population. And this has been modeled to be effective across genera in different fish species. But it doesn't matter if the fish have a ZW, ZZ, or an XXXY sex determination system. The first steps are to be able to feminize fish. And the blue circle indicates what your Trojan sex chromosome carrier is. And so we have to go through steps of sex reversals and selective breeding assisted by genetic sex ID markers in order to produce our Trojan sex chromosome carrier. And so once we produce our YY males, we release them into the system we're trying to manage, and we can eradicate the nuisance population. Same thing with fish that have a ZZZW sex determination system. We release those feminized males and release them into the system. They spawn with wild type males and can eradicate the population. So in our two species, we don't know the sex determination system of these fish. We don't know a lot of information that is required in order to accurately assess the feasibility of using a Trojan sex chromosome eradication strategy for these invasive populations. So there were a lot of questions that we needed answered. So we needed to look at the gonadal differentiation of both species in order to time the estradiol treatment, which would feminize our fish. And so we developed lab-based aquaculture techniques and determined the onset and duration of gonadal differentiation to create effective sex reversal treatments using the hormone estradiol, 17-beta estradiol. And the reason why it's important to look at the gonads of developing fish is we can time our estradiol treatment that will lead to irreversible feminization of males if we do it during the labile period. And so the best way to identify that is through histology. We tracked the gonadal development of males here on the left and females here on the right, and we saw that females differentiate much earlier than male red shiner. So ovaries were starting to differentiate from undifferentiated gonads 2 months before testes were starting to differentiate from undifferentiated gonads. So we identified the labile period to be roughly between 45 days old to 105 days old. And we saw that our red shiner were able to reach sexual maturity by 138 days post-hatch. But we still needed to figure out if there were any genetic differences between male and female of red shiner. And we wanted to try to elucidate what their sex determination system might be. So we employed restriction site associated DNA sequencing, also known as RAD-seq, in order to explore the genomes of male and female red shiner. And so we're using restriction enzymes that chop up the genome. So we have a reduced representation of the genome. We then sequence these fragments and look for polymorphisms between the male and female populations that might be sex-linked, so that we can use them as a marker for who's genetically male and female. And we did this in collaboration with Idaho Fish and Game, and we did bioinformatics using the STAX program and Python. And bioinformatics is where we're just analyzing the sequencing data and organizing it and making sense of all of it. So from our RAD sequencing data with red shiner, we saw the majority of our candidate markers fit an XXXY sex determination system where the male is the heterogametic sex. And we developed 2 sex ID markers that have been effective at predicting phenotypic sex. And so we see the females are homogametic for the allele, so they're CC with this SNP, and they're heterogametic for the allele, so they're CT for this SNP. We did see that some males were already homogametic for that male coding SNP. So there are some females that do in the population that do have that are heterozygous for this SNP, but we're just trying to find a sex-linked SNP. We're not necessarily trying to find the male coding region of the genome, although that would be great. We just need it as a guide to figure out who is genetically male from our sex reversal treatments. And so based off of this genetic research, we see evidence of a male heterogametic sex determination system. And our next steps are to look at sex ratios of progeny from neo-female and wild-type male crosses, and to also use this sex marker to test progeny for SNP sex linkage. So we should see roughly 25% of these fish to be YY males, 50% of them to be heterozygous for the male allele, and another 25% to be homozygous, or just essentially a wild-type female. And then next, I'm going to talk about the estradiol treatments that were necessary to feminize red shiner in the production of YY males. So we use estradiol at various doses from 2 to 120 days post-hatch or 20 to 120 days post-hatch to produce all female cohorts of fish. And we saw that the 100 milligrams of estradiol administered from 20 to 120 days post-hatch produced all female fish. And so we were then able to use our sex ID marker to spawn those neo females with wild type males, and we're also sex reversing some of the offspring from that cross so we could have female YY females. And we did that by, we used pit tags in our sex reversed males and took a fin clip from them to run the genotype, and we crossed the heterozygous females, these neo females, with heterozygous males. And so this is an egg from an all male red shiner broodstock. So this could be the first YY male red shiner ever, but we're still collecting data on this. And now we will be seeing if the SNP marker can identify YY males and YY females from this last cross. So yeah, we saw that right now with the red shiner, we see that there's evidence of a male heterogametic sex determination system, but not until we assess the sex ratio of the progeny from a neo-female cross and a wild type cross will we really be able to say that they're XXXY sex determined. And then we'll also be able to use our sex ID marker in order to isolate YY males from that cross and continue in the production of YY males. With green sunfish, it's been more complicated. So we produced an aquaculture protocol for this species, and we were able to track the gonadal development of green sunfish. And so we see that we also find females differentiate, their ovaries differentiate before males' testes differentiate. But I want you to take note, we have a heavily male skewed population in just our wild type crosses, which is alarming. And so we developed aquaculture protocols for these species and published it in Aquaculture this year. They reached sexual maturity by 213 days post-hatch, which is fantastic because it allows us to do a lot of generations, but we've hit some roadblocks with this species. But we were able to identify the lay-by period for this species, which is roughly 30 to 90 days post-hatch. So let's look at the genetics of green sunfish. We employed the same methods for green sunfish as we did with red shiner. And we saw you can use differences in number of loci, in sequencing coverage, and overall heterozygosity between males and females to be an indicator of which is the heterogametic sex. And so without even a sex ID marker, you can use clues from your sequencing data to elucidate what the sex determination system of your species might be. But unfortunately, we see male and females in our population had very similar number of loci, of roughly the same sequencing coverage and heterozygosity. When we ran our bioinformatic analyses, we found that there were many more ZWZZ markers than XXXY markers, and that might be an indication of their sex determination system. But when we developed our sex ID markers, we saw universal amplification in pretty much all of our samples. So even in species where there was not supposed to be the genetic marker, or in the sex where there was not supposed to be the genetic marker, it popped up in our test. And so with those sequences that were not supposed to be in our in one of the sexes, we see that it did pop up in the majority of all of our fish. So it's not a reliable sex ID marker. So we did a presence-absence marker where a whole sequence was present in one species and absent, or present in one sex and absent in another sex. It keeps saying species instead of sex. And then in our SNP markers, it's the same thing. We saw extremely low concordance in both sexes with the genetic sex ID candidates that we developed. So we've concluded that there's very limited genetic differences between male and female green sunfish based on the high amount of ZZZW sex or sex ID candidates that we got from bioinformatics, that might be their underlying sex chromosome complement, but there could be other environmental effects, and I'll talk about that a little bit later. So my last chapter of the dissertation is going to be on the effects of estradiol on the survival, growth, and sex reversal of green sunfish, because we've yet to isolate any sort of genetic difference between males and females. So we did a similar sex reversal treatment on green sunfish, and we were able to produce all female cohorts from our estradiol treatment group, but you'll see very low percentage of females in our control group. And we also observed a heavily male skewed sex ratio in fish sample between 79 to 322 days post-hatch. The gonads, however, the ovaries in our estradiol treated groups develop normally. They look really great, and they everything as far as the fish development was normal between the treatment groups. But we have this situation where we have a very heavily male skewed sex ratio. And so what it comes down to is we do not have a functional sex ID marker that can guide breeding neo-females with wild type males. We could start just selecting random fish from our sex reversal treatments and spawning them with wild type males to assess the sex ratios, but the sex ratios from our controls are already highly divergent from a 1-to-1 sex ratio that you would expect from a chromosome-based sex determination system. Now, we've seen in their research in bluegills that showed that they have an underlying CZ-ZW sex determination system with increased rearing temperature skewing sex ratios towards males. And we have reason to believe that green sunfish may also have a ZZ-ZW sex determination system, because crosses with green sunfish in other species, the hybrid progeny from those crosses are usually male skewed. And according to Haldane's rule, the sex that is absent in a hybrid progeny is believed to be putatively the heterogametic sex. But we really need to determine what environmental influences are acting on sex determination and sex differentiation in green sunfish in order to elucidate if there is any underlying sex chromosome in that species. So there's more research that needs to be done on green sunfish, especially with regards to different rearing temperatures, different rearing densities, in order to figure out what different environmental influences might be doing to the sex determination and sex differentiation of that species. I want to thank you all so much for listening to my presentation. Once again, if you have any questions, please reach me at chadsteele@email.arizona.edu. Thank you, and y'all have a good one.